Hello, my name is Matthew Usher, and I'm going to be doing a presentation on macromolecules, their monomers, their bonds, their structures, and their functions. The first macromolecule we'll be looking at is carbohydrates. They are made up of carbon, hydrogen, oxygen. The monomer of carbohydrate is a monosaccharide, and these monosaccharides mean single sugar, and these are held together by covalent bonds, and its primary function is as an energy source. Now, the structure of a monosaccharide is right here. It's a ring shape, which can either be in an alpha or beta glucose structure. This means when it's in a ring shape that it is in solution. Fructose is a structural isomer of glucose, and galactose is a stereoisomer of glucose, but they're all monosaccharides. Moving on to lipids. Lipids are primarily composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The, the monomers of lipids are glycerol and fatty acids. The bonds are hydro, are mainly hydrocarbon bonds, which are nonpolar, which are also covalent bonds. The the functions of lipids is to store energy, but they do have many other functions, such as they're very important in making biological membranes, insulation, and also in acting as hormones. Now, we're going to look at the two monomers really quickly and their structures. You can see here the hydroxyl group that's bonded to the extremely long hydrocarbon chain, and they're broken up into saturated fats and unsaturated fats. Unsaturated fats have a double bonded carbon, as you can see right here, right? So we'll be moving on now to proteins. Proteins are primarily made up of carbon and hydrogen, nitrogen, and oxygen. The monomer is amino acids, and the bonds vary depending on the different structure. The function is primarily for cell repair and cell maintenance. Now, the different bonds, for example, in the primary structure would be peptide bonds, in the secondary structure would be hydrogen bonds, in the tertiary structure, hydrogen bonds, ionic bonds, and disulfide bridges, and in the quaternary structure, hydrophobic and hydrophilic attractions play a great role in bonding. As you can see here, this is the general structure of an amino acid, right? And you can see here that they have the amino group, they have the hydrogen and the carboxyl group, and then our group tends to be the variant, which will give you one of your specific 22 amino acids. Now, just going back to the functions, other functions it can also be an energy source. It plays an important role in enzymes, hormones, and antibodies, and it's also extremely relevant in transport. Now, I'd like to move on to nucleic acid, which are made up of carbon, hydrogen, oxygen, and phosphorus. The monomers are nucleotides. The bonds are ester bonds or glycosidic bonds. The functions are basically mostly important in primarily in DNA and RNA synthesis, but other functions would be it reserve bank of genetic information responsible for maintaining the identity of different species, cellular function under the control of DNA, the basic information pathway, and DNA's direct synthesis of RNA, which turns into protein synthesis. As we can see here, we can see the general structure for a nucleotide structure, which is the monomer of a nucleic acid. And then it has a phosphate group and a sugar group, and then the base group right here. And then we can see the different types. You have ad, aden, you have guanine, cytosine, thymine, and adenine. adenine. And these would bond with each other, forming the different links in DNA. And anyway, that's it. I'd like to thank you for your time, and I hope you learned something.